Good afternoon. My name is Haluk Özkaynak, and the topic of my lecture today is health risk assessment. I want to express my gratitude first to Professor Ling Chu Janice Qian for her kind invitation for me to present this lecture for you, and also to Professor Sheng Jasmine Chow for her encouragement on this activity. As you all know, risk assessment is a very important matter for us in our everyday lives as well as in our careers. But at the same time, the topic of health risk assessment is very complicated, and it has many components and incorporates multitude of information from different disciplines. First, I wanted to share with you a brief background about my background. I have a PhD in theoretical physics from Harvard University in Cambridge, Massachusetts, and subsequently a master's degree in environmental health from Harvard School of Public Health in Boston, Massachusetts. I have over 40 years of experience in conducting environmental health measurement studies, exposure analysis and modeling, performing air pollution epidemiology studies, and of course conducting environmental health risk assessments. I was a lecturer at Harvard School of Public Health for many years, and after that I joined U.S. Environmental Protection Agency's Office of Research and Development, heading their exposure modeling program for close to 17 years. In my career, in addition to conducting research projects and teaching, I collaborated with a lot of graduate students, postdoctoral research fellows, and colleagues from different institutions, not only from the US, but also from other countries as well. And in particular, I want to note that I participated in quite rewarding collaborations with researchers from TMU, university faculty, as well as scientists from other institutions in Taiwan. And I look forward to spending the next couple hours with you going over my presentation material. And I believe you have copies of my presentation, so feel free to make notes as I go along. Thank you for your time and attention, and let's start with the presentation. Slide 1 has the specific title of my presentation, and it's Health Risk Assessment with Applications. Two applications that I'd like to go over today. One is environmental health risk assessment, and the other one pertains to COVID-19 health risk assessments. Next slide, please. On slide 2, we talk about health risk assessment, what we mean by that, and what its components are. The definition of health risk assessment is that it's a process that's used to determine the likelihood of adverse health risk posed to either individuals, communities, or population groups of interest from exposures to contaminants or agents of health concern. And the main components of health risk assessment include hazard identification, hazard characterization, exposure assessment, dose response analysis, and risk characterization. And we're going to again demonstrate it with two applications. On the next slide, slide 3, there is a graphical description of how environmental health risk assessment is captured in terms of is relationship between source to adverse effects, what is called the source to effects continuum. The purple squares or rectangles illustrate each of the steps involved in the environmental effects continuum, and they're on the left side of the slide. On the right side of the slide are listed are the measurement data or types of models used to generate information that link these steps together. So for example, the sources of pollution starts from environmental releases, results in environmental concentration, which leads to human population exposures or individual exposures, then the intake dose and target dose, which might result in early biological effects and ultimately to potentially adverse outcomes. And on the next slide, slide 4, I want to sort of provide you a resource for finding out more about the risk assessment steps and the procedures that has been put out in a manual by EPA's Science Policy Council. It's a handbook of risk characterization, and it provides information on the number of issues that I touched upon earlier. On the next slide, I'd like to go a little bit more in detail, that's slide 5, into components of human exposure and risk assessment. This is an expanded version of the figure 1 that we already seen in slide 3. This again starts with source formations, transport, and how the environmental concentrations linked to human exposures through human activity patterns, and then to individual dose and effects. Just want to let you know that there are many types of environmental agents of concern, chemical, microbial, physical, and bioaerosols, etc., and are air allergens, and they're transported in the atmosphere through different mechanisms, as some of them are listed on this slide, like dispersion, distribution, meteorology, etc. And they result in contaminants that we encounter in air, water, food, soil, and dust. And how we're exposed depends on our activity. And that's the key part of the slide that I want to emphasize, because human activity, in terms of where we are, what we do, how much we eat, that also influence significantly the exposures. And then the other issues are the pathways of exposure, the duration of exposure, frequency of exposure, intensity, and timing of exposures are critical factors in determining exposures. And finally, the effects can be short-term acute effects or chronic effects, as the top right of the slide demonstrates. On slide 6, next slide, I list here the likely environmental agents of interest. This is not by any means a complete list of agents that we're concerned about, but among air pollutants, of course, we're interested in fine particles, various gases like ozone, nitrogen oxides, sulfur dioxide, etc. In indoor air, environmental tobacco smoke is a significant concern, as well as biological agents, house dust, mites, and endotoxins, and various allergens. And we're exposed to also metals during our daily activities. And in our homes, we're exposed to pesticide applications that are coming from organophosphate pesticides or pyrethroid pesticides. And also the building materials, for example, contribute to phthalates, organic compounds, and consumer products and other building materials contribute to volatile organic compounds, and so on. So there are a lot of agents and potential pollutants that we might be exposed to. This sort of reminds me of this saying that's often cited, originated from in the 15th century by Paracelsus, that dose makes the poison. However, exposure makes the dose, because if you're not exposed, you don't get the dose. And of course, depending on your exposure and dose, that determines the severity and the degree of the adverse effects. So that's another reason why we have to deal with pollutant-specific and pathway-specific exposures to be able to tease these connections out. On the following slide, I'm switching a bit to issues regarding children's environmental health issues. Slide number 7. And again, these are some examples, but not all. So for example, asthma and immunological effects in children caused by exposures to some of these toxic agents, especially like metals and pesticides, developmental disorders and autism, neurodevelopmental effects, cancer, acute poisoning, etc. Now I want to show you very quickly some slides how children's exposures might be occurring in the real world in terms of various pathways of contact. On slide 8, we see a possible exposure pathway for kids, in this case 2 different kids. On the left is a small girl, a toddler, trying to reach to a toy on a carpet, but we don't know exactly what's on the carpet, and the kid is crawling on it, getting stuff in her hands, and then subsequently to her mouth during hand-to-mouth activities. On the right, an older child is playing on an old play set, an older play set, I should say. And these type of play sets used to contain CCA material, which had arsenic and chromium. So by getting, again, in terms of dermal contact, the kid can be exposed to a highly carcinogenic compound. On a side note, these 2 pictures are children of 2 of my colleagues, and they're both gotten very old since the pictures were taken. One just started college, the girl on the left, and the boy is now working on his graduate studies in epidemiology. Now the next slide, we're going to look at object-to-mouth contact. And again, this is the same girl. Here the concern is the kid has innocently placed a rubber ducky in her mouth, and the concern there is phthalates in rubber ducky are toxic. And this is another pathway that the kids might be exposed to, not just rather hand to mouth, but directly putting an object into their mouths. And in the following picture, I'm going to show an example of ingestion and dermal contact together, and that's slide number 10. Here the kids are playing next to a fountain outside in a park somewhere, and the concern there will be what's in the water. And that's been a big issue these days in the U.S. because we have cities in Illinois, in Detroit, have contamination from lead, and in other areas in arsenic that are possible toxic agents that are, well, arsenic is known to be a carcinogen, and lead is a neurotoxin. So, again, that's another example. And the final example of a pictorial example is the next slide, slide number 11, dietary and non-dietary ingestion. Here the kids are eating their lunch near their playground area and enjoying the food. Of course, the food, depending on the type and where it came from, can contain some of trace amounts of these various agents that I mentioned earlier. And non-dietary ingestion is when kids play on the grass or inside the house and ingest soil or dust in their mouths. And that is actually being found to be a very important determinant of risk assessment, risk assessments conducted for Superfund sites where there's contaminated sites, and then the highest risk has been found to be the incidental soil dust ingestion for kids. So, next slide, I'm going to be talking about how we go about measuring exposures, even though I'm not going to be focusing that in my presentation today, but I just want to at least make you be aware of. There are a number of ways of we go about measuring exposures in community health studies. Typically, there is no single exposure methods that can be used like a cookie cut type to characterize individual exposures in conducting a community health study. Measurement methods can include environmental samples, personal monitoring, biological samples like blood, urine, hair, etc., questionnaires where people fill out forms in terms of what they've been doing, what they've been coming in contact with, etc., and time activity diaries are very important, especially for models, in terms of getting an accurate information when and where the kid was or a person was at a given day, so that the right amount of information can be linked together to generate their potential exposures. Of course, in trying to decide which one of these methods to use, we have to consider a number of things. Their strengths and weaknesses, method difficulties, how burdensome they are, how invasive they're going to be. The cost involved is a critical determinant. How often you have to measure, what exposure period you're going to characterize, etc. Next slide, please. On slide 13, I alluded to earlier to use of exposure models and why they depend on time activity information. And here I'm going to be talking a little bit about the definition of exposure models. Exposure models are scientific organization and interpretation of complicated human exposure scenarios. So they try to characterize realistic routes and pathways of exposure for individuals or populations. And they also estimate physiologically relevant time periods and durations, what the exposures are. And they identify what activities and behaviors are related to exposures and uptake rates. And I'll be showing some examples of these shortly. And then they interface with the physiologically based pharmacokinetic models, which are PV-PK models, to provide actual exposures and dose for different age and gender sensitive groups. So they serve a critical link between emissions, concentrations, exposures, target tissue dose, and toxicity information. The next slide, I'm going to talk about briefly the types of exposure modeling methods that have been used so far. On slide 14, the first of those models is listed as statistical models. They employ empirical data from personal monitoring studies. So they've looked at personal monitoring information and look at the measurements indoors or outdoors and try to find statistical relationships between those measurements. So in light blue, there is a simple regression model written here for estimating inhalation exposures as an example. So exposure equals a constant term plus regression coefficient times indoor concentration and how much outdoor concentration penetrates indoors. And then the third term is the amount of outdoor concentrations that go out. So we have microenvironmental exposures characterized using statistical variables. The second model, meteorological models, are the best examples of these, or air quality models, and a lot of the pharmacokinetic models are deterministic or mechanistic models. So they start from the first principles and look at the processes that provide exposures, concentrations, or exposures. And a third model, an area that I've worked a lot, are the stochastic models. And they're sort of extensions of the deterministic mechanistic models, where rather than using constant terms as inputs or parameters of the models, we use distributions, not single values, to describe the variability and sometimes the uncertainty in the information that goes into those important inputs or parameters. So consequently, the model, as a result, generates variability and uncertainty in the results, since it incorporated variability and uncertainty in the data that it utilized in the model runs. The popular method that is used is Monte Carlo simulation model or random sampling techniques, and the output distributions can be either variability distributions or uncertainty distributions or both of them. On the next slide, slide 15, I show what type of human activity diary information that are used typically in these stochastic models. The main information that comes for most of these models are from EPA's Consolidated Human Activity Database. In short, acronym is CHAD. And this is available to the public, and if you Google it, you'll see the links to it and necessary documentation and publications. It's a compilation of several human activity surveys, which are sort of shown on the right side of the figure, which we're not going to go into the detail now. The information includes, of course, demographic information, age, race, and gender. And the diary data, however, is standardized format. It's over 24 hours, 1 minute to 1 hour increments of each activity, location, or type. And what that means is that if a person spends, let's say, 3 hours indoors, just reading or resting, whatever, the activity period will be 3 hours. But a person just goes in and out of the house or into the car, the activity levels can be of the order of a few minutes or even as short as 1 minute. So the typical range is from anywhere from a few minutes to 1 hour. And the diary database allows for 114 locations and 144 activity types. So it's a big database. In this slide it says 22,968, but it's been more recently the database have increased to close to 50,000 diaries because it incorporated other than the EPA-sponsored studies now. So what is the limitation of this database? The limitation is, and there's something that I touched briefly, is that it only provides 24-hour diary information for each participant. So this is an issue for trying to model an individual over a long-term period. If you want to know chronic exposure over 365 days, this diary database does not provide, except in few cases for maybe 2-3 day periods, longitudinal data or multi-day diary data. So you have to make some assumptions about what a given individual representative simulated individual might be behaving in the following days. So the next slide, slide 16, shows how this information is used calculating individual exposure profiles. So on the top left, we have the child diary. We have the location, activity, minutes, and something called METS, which represents the energy expenditure that's mostly used for getting breathing rate information, depending, for example, if somebody is jogging, they're breathing more heavy. If they road jogging, so the METS will be a higher number. And then on the bottom, on the left side, is of course, the concentration information, which is the driving force, and giving you 3 typical examples of distributions in red. And then multitude of exposure and dose factors, which are shown on the gray. And that relates concentrations to exposures depending on how much these concentrations get on the person through which exposure route and process. And the output will be actually, in a stochastic exposure model especially, is separately predictions for inhalation exposures, dietary ingestion exposures, dermal exposures, non-dietary ingestion exposures. So let's look at the dietary ingestion here on the right side, the second green one. It shows the person had a breakfast, lunch, dinner, and had few snacks here and there. So the exposure occurred during that time. Inhalation exposure, the person wakes up, goes around getting ready, and then goes to work, maybe goes to school. The exposures go down a little bit, and then after the day is over, the school is over, the person starts getting more active, so the exposures go up. And similarly, the dermal exposures depend on the activity and varies during the day, where that activity is taking place, and non-dietary ingestion the same way. And on the next slide, I want to show you the main components of the stochastic exposure model that I worked on with my colleagues at EPA. It's called the SHEDS model. That stands for Stochastic Human Exposure and Dose Simulation Model. It incorporates both variability and uncertainty. So what is variability? Variability is temporal, spatial, or inter-individual differences in the value of an input. For example, body weights vary from person to person, body heights, etc. And these are considered to be variable inputs or parameters. So when we incorporate these information into the model, we get a distribution that shows the percentiles of predicted exposures and dose. So the exposure and dose is in the x-axis and percentile in the y-axis. Now we know that we're not certain in terms of choosing these values, in terms of what the mean body weights are, what the mean transfer rates of material are when a person puts their hand. Depends on the moisture, depends on the skin characteristics, depends on the soil characteristics, etc. So these are all referred to as uncertainty. So the measure of incompleteness of our knowledge is considered uncertain. So the stochastic exposure model, this takes a lot of time and information to generate these results for uncertainty, provides multiple variability distributions depending about the uncertainty of the variables. So on the vertical side is the uncertainty percentile, and on the horizontal side is the uncertainty about exposure or dose for a given percentile. So if we look at the x-axis, the exposure dose, if we pick a number, we see that that number can be as low as the one for a given percentile at CDF1, way on the left, or very much on the right, depending on which percentile we choose. So, and then you can do the same thing about a given dose. What percentile will that be? So if we look at a higher dose, the one on the right shows a lower percentile, but the ones in the middle and the left will show a much higher percentile. So the uncertainty is very important, but unfortunately at the same time, it's not easy to characterize, and also depends on how many studies are available to be able to determine the information for that. And so how is this done? In general terms, is shown on the next slide. Slide 18 shows the Shed's model structure, basically three main components. These are the input databases that I mentioned earlier, the census data, human activity data, the chat diaries, ambient concentrations, food residues, and if we're dealing with diet, recipe information, etc., to be able to determine what ingredients are used in making, let's say, an apple pie, so that you find out how much sugar goes into it, how much flour goes into it, what's in the flour, what's in the sugar. So it's actually dietary exposure modeling is fairly involved and complicated. But there are databases that have been developed by EPA and other agencies to support that type of calculation. Now the algorithms, of course, depends on the pathway of exposure, the route of exposure. The inhalation algorithms, of course, depend on the breathing rates, and they're somewhat fairly standard. Ingestion depends on quite a bit in terms of what time people eat and how much they eat, what kind of food they eat. And dermal information is also very complicated because it depends on the age of the subject and the activities of the subject. For children, I mentioned crawling, playing on surfaces that are contaminated, like in indoors in terms of rugs and carpets that have dust and soil, trapped in soil, that have metals and pesticides, and outside in the grass. Also, of course, these same and other chemicals, including organics, are involved. How much do they touch the surfaces, and then how much after touching they put their hands into their mouths also creates the ingestion information. So the dermal contact is first characterized, and then hand-to-mouth ingestion is characterized in that situation. And the outputs are similar to the previous slides, in terms of various distributions. The Sheds model produces on the top right, on the right-hand column, distributions for exposures from each of these three pathways, and then it sums up as a total exposure as well. And then population dose through either a straightforward absorption factor or a PBPK model to convert the exposures into a target dose. Okay, next slide is, I already said quite a bit about it. Number 19, slide number 19 is the dermal exposure profile. This is an example, real example of a, based on a child's diary. The child starts his or her day around 10 o'clock, playing on texture surfaces, smooth surfaces like sofa and couch indoors, and then, does a hand-to-mouth activity which lowers the concentrations in the hand or on the skin surfaces. And then around 4 o'clock or so goes out and plays on the lawn, and it gets exposure to potentially a pesticide. And then these playing outdoor exposures stop when the child gets indoors, and at nighttime we assume the child takes a bath, that's the water here, and that provides the removal of the soil and dust and the associated contaminants from the skin. The next slide, very briefly, slide 20, shows the dietary ingestion profile for a typical individual. Again, like I alluded before, there's the breakfast, lunch, dinner, and snacks. And of course, each person is different. So if you look at the different diaries, you'll see a totally different picture. If you look at my diary, you'll see a different picture than this one, because I'm a little late in eating my lunch than the normal person. So those have to be incorporated when doing a large population exposure modeling using the CHAD diary databases. Now I want to give another example. This is a real example that relates to that slide that showed a picture of a kid playing on a arsenic treated play set. And this is a project that I worked on a number of years ago, actually, with Professor Winston Dank, who's been with TMU, but at that time he was with US EPA, Office of Antimicrobial Division. And they asked for our assistance in evaluating the risks associated with the chromated copper arsenate treated play decks. And we worked it a number of years, and then based on our findings, actually the manufacturers voluntarily banned the use of CCA in play sets and play decks at homes. And if you go to the playgrounds these days, you don't see these kind of wooden structures replaced by plastics or other materials. So the slide 21 shows that the variability of the distribution of predicted arsenic dose for children playing on a play set or a deck is around 10 to the minus 5. That's the variability distribution. But we have done an uncertainty distribution associated with our runs for this case. And if you see that the top curve represents our 95th percentile predictions of our simulations, and the lower curve presents the fifth percentile, the lower concentrations and dose that are predicted for this scenario. So with that, I think we're going to switch to dose, the second component that's important to the definition or characterization of risk. And slide 22 shows various complicated dose response curves. These are, depending on the agent of concern, depending on the experiment, one will see a lot of different dose response curves. For example, the one on the left, shown in red, for most of the low dose concentrations, it's a linear type of a curve. Whereas if you look at the yellow, black, and the blue, the ones in the middle, they're more sort of have a threshold and then start gradually rising, and then some saturation perhaps at the very high levels. So these dose response relationships are important in terms of determining the potential cancer or non-cancer effects, which we'll be discussing pretty soon. So on the next slide, 23, I want to get back to the uncertainty here. In terms of the uncertainty that involves how to utilize information from data based on experiments conducted on other species to human populations. First of all, there's a variability. Humans are not all the same. There's individual to individual variability. The uncertainty factor associated with that is described as UFH, H for human. And then animal to human extrapolation, like mice to rat extrapolation is my example. So that as a subscript of A. And subchronic to chronic extrapolation, because studies are not necessarily done obviously for the entire length of the subject or the animal. So that's also ascribed an uncertainty factor. And then the other one is a little subtle one. When the experiments are conducted, they can identify sometimes no adverse effect levels, but sometimes they can't. They identify a low adverse effect level, so they do not get to the point that no adverse effect level is obtained. So that has to be extrapolated. So what is called LOL to NOEL is the extrapolation for that, and that has a subscript of L. And above all, and across all of those elements, there are the database deficiencies. So how robust are the data? Do we have multiple studies? And the sufficient number of subjects or animals or experiments have been conducted? So that is also ascribed an uncertainty, UFD. D meaning database. So the final composite or total uncertainty will be a multiplier of all these uncertainty factors. UFH, UFA, UFS, UFL, UFD. And so typically the uncertainty factors range from 1 to 10, depending on the quality and the quantity of our knowledge. So if the knowledge is limited, so we have 5 orders of magnitude uncertainty. So each U gets 10, so 10 times 10 times 10, 5 times 10 is 100,000. So the uncertainty factor can be as high as 100,000. So that's how the risk assessment assures that the safe levels for humans are obtained in a conservative way, meaning more protective way. And the next slide shows an example of how this is actually applied. That's slide number 24. There is a dose response curve that shows low L, the lowest observed effect level, which is around, I'm guessing, 3 or 4, and the no adverse effect level, which is of course at a lower concentration. It's somewhere around 0.5 or something like that. So the uncertainty factors are multiplied on each one of these, and the minimum risk level that's determined by multiplying all these uncertainty factors that are associated with NOEL and LOL are shown to be on the very left at 0.00001 level. So that's how much safety is incorporated when we're defining a minimum risk level in risk characterization. Now, let's talk about how the exposure dose analysis can be conducted in a deterministic fashion, so that we get a somewhat easier sense of understanding how this is done. For example, the slide 25 shows the equations involved in determining potential dose from exposure to soil material. So the numerator of the equation involves 4 factors, and since there wasn't enough room here, the presenters have written them over each other, but they're all multiplied together. You start with the concentration in the soil, the intake rate, the exposure duration, and the conversion factor that converts from kilograms to milligrams and other way around, divided by the averaging time, and typically for adults that's about 70 years times 365 days, so this is in days, and then the body weight of an individual. So the dose units are milligram of substance per kilogram day. And how much of that is observed is obtained in this case by a simple fraction. This is called the absorption fraction, that multiplies the potential dose shown above with that factor. The concentration for soil is milligram per kilogram. But if you're talking about air exposures, the equation is not given, so I'm just sort of cheating here. Just trying to give you a sense. It has to be micrograms per cubic meter. If it's water, it is milligrams per liter. Then intake rates are meter cube per day for air. Most adults breathe about 20 cubic meters of air per day. And in terms of the water we drink, that varies. I guess on the average it's 1.5 liters per day. And averaging time, as I mentioned before, it's 70 years times 365 days. Well, on the next slide, which I'm not going to go into too much detail, we'll talk about definition of exposure variables. The intake is I, or dose is D, and defines the measure of milligram of the chemical entering the body per kilogram body weight. That means milligram over kilogram per day. Concentration we talked about in terms of its units. Ingestion or inhalation is the rate at which chemical is ingested or eaten or inhaled per day. milligram per day or meter cube per day. And I would like to give a soil ingestion intake rate as an example. It can range from anywhere from 20 milligrams to 100 milligrams of soil and dust ingestion for kids. And that includes the work done by many researchers, including Professor Chian here at TMU, as well as my group at EPA, and others that worked on this project from other institutions as well. So the exposure frequency is the measure of events per hour or day. Exposure frequency is measuring the exposure days per year. And exposure duration is the amount of time a person is exposed. For example, hours, days, or years. That's important for people who have occupational exposure potentials because they're not exposed 24/7, but they're exposed potentially while they're at work. And I mentioned the averaging time, the period over which exposure is averaged, measured in days. The next slide provides an example of the values used for some of these exposure variables. That's in slide 27. These are the recommended default exposure factors for those calculations by the Agency of Toxic Substances and Disease Registry, and the information is 7 years ago now, or more than that, 8 years ago. But nevertheless, it is fairly informative. If you look at the left panel, which I like to draw your attention, is that the body weight varies quite a bit depending on the age of the child. When the kid is under 1 year old, weighs about 8 kilograms on the average, and then 11 kilograms between 1 to 2 years, and when the kid is 6 to 11, goes up to 32 kilograms. These are again averages, and when it reaches age 16 to 21, it's up to around 71. And adults older than 21 years old are ascribed 80 kilograms. Actually, when I was a student, that number was 70 kilograms. That represents the shift of increasing body weights in the populations in many countries, especially in the United States. And then the right side of the panel gives the default values, exposure frequency, exposure duration, and inhalation exposure. And it also, interestingly, on the very right column is that it sort of, in some cases, provides the default as either as a reasonable maximum estimate, as RME, or a central tendency. So these are important in terms of interpreting your results from your risk characterization in terms of which one of these numbers do you use. For example, exposure duration for residential scenario, they provide 33 years as a reasonable maximum estimate, and central tendency is 12. The reason being is that they've found in population surveys that people move from one house to another on the average of 12 years. And I guess they found out the reasonable maximum levels of people who have been staying in the same home or location is about 33 years. In the same way with the occupational exposures, there are people who stay short term at one job versus longer term at another job, 25 years versus 6.6 years. Next slide shows the suggested source or recommended source of risk information, where to find information that links the potential health outcomes resulting from exposures. And the well-known source is the EPA's Integrated Risk Information System. In short, it's called IRIS. IRIS is a human health assessment program that evaluates risk information on health effects that may result from exposures to, it says 500, but now it's more than 600 or so chemicals, contaminants. And it's the main source of route-specific cancer and non-cancer risk reference concentrations or dose values used in health risk assessments. For example, if you calculated a person has an exposure such and such levels, based on all the considerations that we talked about, what are their chances of coming down with cancers or some other non-cancer morbidity outcomes. And this information for over 600 chemicals are described in detail on EPA's IRIS database. And the website is provided here for those of you who have not perhaps looked into it. And IRIS also provides hazard identification and dose response assessment information for a large group of chemicals. And I provided you the website for that. If you go to the main IRIS website, you can get to all these various data sets that the IRIS database has, and this one is especially for chemicals. So if you type in benzene, for example, you'll get all the information, what the benzene-associated cancer risks are for a given unit concentration. So this information then be combined with exposure information to characterize public health risk for a given substance in a given situation. And as I mentioned above, the non-cancer effects are also characterized in terms of RFDs or RFCs, reference doses or reference concentrations. Those are the terms that it stands for. And in most instances, RFDs and RFDs are developed for non-carcinogenic effects of substances. And finally, cancer effects describe the weight of evidence for human carcinogenicity, oral slope factors. I'll show you an example of how we apply that, and oral inhalation unit risk for carcinogenic effects based on using linear, no-threshold statistical risk estimation methods. It's like that earlier figure that I presented with the red line. They're assuming a linear relationship close to the no effect level at the zero concentrations and the higher levels. So carcinogenic risk assessment assumes a linear, no non-threshold effects. Next slide, we're going to be talking about how the equations might look like for non-cancer, and then after that, cancer effects. How do we characterize using the information from IRIS and based on our calculations of reference doses and exposure average daily dose? What is the potential risks associated with that? Let's look at the non-cancer effects, slide 29. The non-cancer effects, I'll start with the right-hand side of it, which is a little easier. For inhalation exposures for chemicals that are airborne, we look at exposure concentration in air, which in this case used as milligram per cubic meter, divided by a reference concentration that's obtained from iris to obtain a hazard quotient. And all other routes use average daily dose for oral or dermal. And the reference value that's used is RFD, the reference dose. It's not a concentration, it's a dose. And basically you divide your estimated or predicted average daily dose by the reference dose that you obtained from iris or some other source. By the way, I do not want to be EPA centric here, because in California, Department of Health has also their independent databases and evaluations for chemicals in terms of characterizing the health risk associated with that. And some of them are actually more stringent than the EPA's, federal EPA's iris values, and some of them are actually values that are not even in iris. So I would urge you to also look at other sources than iris. California is a good one, and WHO is another one, but WHO is sort of incorporates information from multiple countries and is heavily weighted by values from the United States, as well as the utility of that information is not necessarily as clearly recognized as the ones from iris or California. The cancer risks are the next in line, which is slide number 30. Cancer risk for oral cancers is calculated in terms of lifetime average daily dose, LADD, times an oral slope factor, again from either from iris, California, or WHO. And the units are LADD is milligram per kilogram day, as mentioned before. The slope factor is just the inverse of that, milligram over kilogram per day. So that gives you a unitless number. And the cancer risk for inhalation for airborne pollutants is lifetime average exposure concentration, microgram per cubic meter, times inhalation unit risk, which is 1 over microgram per cubic meter, or extra risk divided by microgram per cubic meter. So the resulting number will be an extra risk. All right, so how do we sort of look at all this information in just one example to get a better feeling for how to use that information? This will be on the next slide, slide 31. Now I pose a problem. What is your lifetime cancer risk if you live in a large urban area that has 1 parts per billion of benzene in the air? Now risk, we just gone over that, is equal to exposure times inhalation unit risk. So the inhalation unit risk, if we look at iris, benzene inhalation unit risk is given as 2.2 times 10 to the minus 6. 2.2 times 10 to the minus 6. But we have an issue here because our units are not consistent with the previous slide. We need to convert parts per billion, which is volume to volume, to micrograms per cubic meter. The conversion is concentration in micrograms per cubic meter is equal to molecular weight of that substance times concentration given in parts per billion divided by 24.5. Now, where do we come with 24.5? It's the volume in liters of a mole of a substance in gram molecular weight of a gas at 1 atmospheric pressure at 25 degrees Celsius. And this comes from ideal gas law. So, then we can go ahead trying to sort of apply these factors to get the right units to be able to calculate the estimated excess cancer risk. So benzene has a concentration of 78 grams. So using that above equation that we just did, 78 divided by 24.5 gets it into units of microgram per cubic meter, times the IRS factor 2.2 times 10 to the minus 6 per microgram per cubic meter, gives you a number of 0.7 times 10 to the minus 5, or roughly 10 to the minus 5 lifetime cancer risks, or 1 in 100,000. So if you're in an urban area that has 1 ppb of benzene in the air, you have a risk close to 1 in 100,000. Now, for pollutants that are in particulate matter form, the concentrations again go in terms of weight by weight or parts per million, which is milligram per kilogram is equal to parts per million. And in this context, ppb is volume by volume basis, as I mentioned earlier. Now, I want to now go to the hazard quotient information and see how that's used in the cancer and non-cancer analysis in the next slide. That's slide 32. Hazard quotients and interventional risks from all exposure routes are estimated and summed for each chemical. So if one is exposed to not just benzene, let's say toluene, xylene, and all sorts of other chemicals in the air, you have to calculate separately for each chemical and each exposure route their hazard quotients. The hazard quotients and incremental cancer risks for the individual chemicals are then added to calculate the total hazard index, which is called HI, and that's the total risk hazard index. For screening assessment, the default assumption is that hazard posed by individual chemicals are additive. However, if there is some non-toxic cancer effects of chemicals that are unlikely to be additive, then a statement can be made that we're not adding those based on target organ or mode of action in our analysis, rather than go ahead and combine different non-cancer endpoints together. Now, in the final analysis, what is done in terms of the regulatory environment? This is notes. Typically, when the hazard index that's less than 1 is considered as an acceptable risk for chemicals associated with non-cancer health risks. And for cancer risks, historically EPA considers cancer risks ranging from 10 to the minus 4 to 10 to the minus 6 to be acceptable. So depending on the program, depending on the context, depending on some other factors too, economics, etc., the EPA's acceptance of a cancer risk can vary between 10 to the minus 4 to the 10 to the minus 6. Now, this is typically a U.S. Situation. It doesn't apply to other countries. And in Europe, actually, they even take a different approach, what is called a precautionary approach. If they think that they have some data that they have a concern about a chemical, they will ban that chemical. And that's a pretty strict way of approaching it. But that's also the most, of course, health protective way. And recent example of that, actually, if you've been following the news, is the perfluorinated compounds, PFAS. Europe decided to ban that, and EPA is still evaluating it. All right. Now I want to talk to something that's probably important to many of you who are going to be medical students. And there are other risk measures that are of interest to other disciplines. And because of my time limitation today, but as well as this material crosses a bit to epidemiology, I did not want to dwell too much on that, but at least alert you to that. And slide number 33 talks about these common risk measures, in addition to the excess risk of cancer and non-cancer, as we discussed. Now, in medical sciences, you see often two other measures that are used. One is RR, that stands for relative risk, which is a risk of a particular event in an exposed or experimental group relative to that in the non-exposed or control group. So if the relative risk is greater than 1, it represents higher risks in the exposed group than in the control group. Now the other one is odds ratio, and I've been seeing that more and more in the medical literature, and that's odds, or odds stands for probability of an event in an experimental group relative to the odds in a control group. For instance, odds of lung cancer among smokers is about 14 times the odds of lung cancer in the non-smokers. Now, how do you calculate these relative risks and odds? It's actually very simple, and if you take epidemiology, which I urge everyone to do that if the curriculum permits that, is a 2x2 table. You have one column exposed, the other column not exposed, and across is observed effect or not observed effect. So you just put the numbers in there. It's just like 4 numbers, A, B, C, D. And if you actually look at any epidemiology book or even just Google, very easily you can find out how to calculate these relative risks and odds ratios using those experimentally determined A, B, C, Ds from clinical studies or other population-based studies. And finally, the attributable risk is a proportion of exposed subjects reporting an adverse outcome minus proportion of non-exposed subjects reporting that adverse outcome. So we're really attributing that risk to that fraction of the people in this manner. Well, this has been a quick tour of environmental health risk assessment. Now this concludes our first part of our lecture. I think we all need to take a short break here. Let's take a brief break of 10 minutes before we start with the next application. Thanks for your attention. Hello. Hope you get a chance to stretch your legs and take a mini break, and ready to resume our lecture presentation. Now in part 2, we're going to be doing something considerably different. We're going to be dealing with a current problem that is of concern to the entire world. That is the issue of COVID. So in part 2, I thought I'll touch upon a number of things dealing with SARS-CoV-2 virus, coronavirus. And slide 35 gives the title of the section, which is evaluating exposures, characterization, and management of risks posed by the SARS-CoV-2 coronavirus. The next slide talks about the coronavirus entry of the body and symptoms in the COVID disease. And many of you are, I'm sure, very familiar with that. A number of you probably have come down with COVID-19, and I know a number of people in my family as well as others have been touched by COVID-19, fortunately not seriously. So the illness of the coronavirus starts with the droplets from infected person through the person coughing, sneezing, or breathing. And those droplets could be air or on a surface that you touch before touching any of your membranes, such as eyes, nose, or mouth, that gives a virus a passage to the mucous membrane in your throat. Typically, within 2 to 14 days, your immune system may respond with symptoms, including fever, cough, shortness of breath, fatigue, chills, body aches, headache, sore throat, which is common, congestion or running in the nose, which is common, and less common, well, it's also still prevalent, is loss of taste or smell. Nausea, vomiting, and even diarrhea have been reported. Now the next slide, slide 37, shows a schematic diagram of the human respiratory tract so that we can get a little better understanding of where these particles or aerosols go in the body after they enter through the mouth or the nose. Now the body's, the respiratory pulmonary system is divided typically into 3 parts. The upper area, extrathoracic region, or nasopharyngeal region, those 2 terms are used, describe the area through the nasal passages, and the pharyngeal region, the nasopharyngeal region below that. And then going deeper down, you get into the midsection, which is the tracheobronchial region, and within the branches of the bronchi, we have the alveoli, which are the small sacs in the pulmonary region, which is shown in the bottom within a big circle. And so how does the deposition, how much of the deposition occurs in various pulmonary regions, the 3 regions we talked about? That's described on the figure shown on slide 38, the next slide. The extrathoracic region, basically most of the large particles that we breathe get captured through interception, basically, because of their size are large. And as the particles get to the smaller size, then they start penetrating more deeper into the tracheobronchial region, but more important to the pulmonary region. So that's the region of concern because that's where our oxygen exchange takes place in our alveoli. And the peak deposition occurs around 0.01 micromillimeter aerodynamic diameter, or about 10 nanometers. A typical coronavirus is about 0.1 microns in size, just for a reference point. So in the next about probably 4 slides or so, or 3 slides, at least 2 slides, maybe, yeah, 4 or 5 slides, I borrowed information from Dr. Sui Huang, who's a well-experienced and well-educated systems biologist who's with the famous Institute of Systems Biology at Seattle, who at the beginning of the pandemic actually posted on the internet in a cancer-related website what happens to the aerosols that contain the coronavirus when people breathe them out or start coughing or sneezing, and the efficacy of the various ways of preventing their dispersion as well as breathing them in through the mouth. So slide number 39, the next slide, shows that most of the aerosols are greater than 10 microns, but if they're in the larger size, which are the bigger red circles, they drop down because of their size and weight in terms of gravitational settling very quickly within 1.5 meters. But the fine aerosols disperse in the air, so they can go stay up suspended in the air for a longer time. But if the person who is infected person who coughs, for example, with velocities of 10 meters per second, even the largest particles can go up to 2 meters, which is roughly 6 feet. But if a person sneezes, the velocities have been reported to be actually in the literature around 50, even to 100 meters per second. Then they can travel all the way up to 6 meters and beyond. So there are a number of factors that determines how far these infected particles can travel from an infected person, depending on whether the person is just breathing normally or exhaling normally or coughing or sneezing. So pulmonary deposition of cough and sneeze droplets, how that happens is shown in the next slide, slide 40. The cough, sneeze droplets reach beyond 6 feet, as we mentioned. And in nasopharynx area, these particles deposit. The ones that are larger stuck in the upper respiratory tract. Much smaller particles, smaller than 10 micron, reach down the lower lung. And that's illustrated with tiny red dots in the alveoli and the bronchi leading to that. So Dr. Huang also shown that the larger particles penetration into the upper respiratory tract can be blocked by surgical masks. But if you wanted to block the deposition of smaller particles into the alveoli, deeper into the lung, you need N95 filters. So they're blocked more efficiently by N95 filters. Now on slide 41, we show some pictures of different masks that you're well aware of because in Asian countries people wear masks diligently, unlike in the Western countries before the pandemic. So the types of masks used for protection against coronavirus include homemade or fashion designed cloth masks, which is on the left. In the middle you see a picture of a surgical face mask, and the one on the right is a N95 respirator mask, which is obviously the most efficient, but it also requires proper fitting. Now, proper fitting is an important issue. This has been known well by industrial hygienists even much before the pandemic. These masks are only efficient to the degree that they can protect you if the mask fits properly. And the usual areas of escape are below the eyelids, or around the below the mouth and the neck area. So the large factor that determines the efficacy of these masks are also linked to how well they fit. So that should be something that paid very serious attention to. So Dr. Huang, in the next slide, shows how much these various types of masks offer some protection to the person in terms of not being able to release the infected particles, as well as how much of a risk they provide to other individuals as the particles leak through that mask. So he uses 3 examples, and these are just illustrative purposes. There's quite a bit of variability, and we talked about variability and uncertainty before in the first part, so these numbers are sort of ballpark figures, bottom line. So he used 3 examples. One is a cloth mask. He says T-cloth, but that stands for cloth, dishcloth, or regular cloth. I don't know whether it's a British terminology or what. If you wear one of these masks, if the reference value of the number of particles, let's say you had 100, 33% or 33 of them will be leaking through that cloth mask. If you have surgical mask, the protection is slightly better. It goes down to 25. But if you have an effective, very effective N95 mask, it's really highly protective. It saves basically 99% of them to be released in the environment. So the particles leaked are out of 100 is 1. So what's the flip side? The other side, I should say, the person that are exposed, particle produced by coughing. This is now is an important factor. So they are obviously coming out with a higher velocity and be able to penetrate through the masks. So if you have a cloth mask, and this is the right side of the slide number 42, the cloth masks does a poor job, about 90% leaks, and the surgical mask provides about 50% protection in terms of the leakage, and N95 actually provides a 30% protection. So, so the protection is not necessarily equivalent to the way that the masks allows particles coming out versus stopping them. So now what happens after a person gets hit by these coronaviruses? That's the next slide, number 43. Again, everyone is familiar by now in terms of the discussions that go on. They've been going on for the last 3 years about the coronavirus and how it gets into the human body, and etc. This is a sort of a author's rendition of a virus that looks like a sphere. There's a viral genome at the center, and then a lipid member around it, and then the spike proteins that looks like a triangular rocket-shaped tops to them, and they seem to match the human cells ACE2 proteins, which have these kind of receptors that accept these kind of spike proteins that come from the SARS coronavirus. So in the next slide, we'll look at number in slide 44, the most likely pulmonary sites for the coronavirus upon entry to human respiratory tract. Most likely site of infection, because of the cough and sneeze droplets that reach 6 feet or so, is actually in the nasopharynx region. Highest is in the nose, and also, of course, the nasopharynx is tied to the oral cavity. And that is why most people experience sore throat, runny nose, and ultimately cough. But the sore throat and runny nose seems to be a most common symptom, even with the variations on the virus that we have seen so far. And then, so the particles larger than 10 microns stuck in the upper respiratory tract, and as we mentioned, can be blocked by surgical masks. But the smaller than 10 micron aerosols can reach down lower lungs. So that includes the bronchi and the alveoli at the end that stick out for us to be able to get oxygen exchange. So they can be blocked efficiently only by N95 filters. The efficiency numbers, again, is dealing with the coronavirus and all that, but if for some of the surgical masks or cloth masks that have been tested before pandemic, has actually seen that depending on the manufacturer and the type of mask, the cloth mask efficiencies can vary anywhere from, let's say, 30 to 70% for fine particles less than 2.5 microns. So again, this should be taken with a grain of salt. But having said that, we know that N95 masks are the gold standard for protection at this very moment. So we talk again on the following slide what the coronavirus does to your body, and in terms of how to get into the various regions of the pulmonary tract. The virus moves down the respiratory tract, namely the mouth, nose, throat, and it gets to the lower airways, where we have more ACE2 receptors than the rest of our respiratory tract. So COVID-19 is more likely to go deeper than the viruses like the common cold. That's unfortunate. So that's where also we have more ACE2 receptors. Lungs might become inflamed, making it tough to breathe. This can lead to pneumonia, an infection of the tiny sacs called alveoli inside the lungs, where we exchange oxygen with carbon dioxide in our blood. If you did a CT scan of a person that has these kind of symptoms and conditions, we see patchy areas called glass opacity in the lung where the damage starts occurring in the alveoli. Again, most people with symptoms end with a cough and a fever, and within 8-10 days things get milder or better. In some cases the infections get severe, especially in the alpha and the delta and beta version of the original variants. We had a lot of serious situations in terms of symptoms beginning 5 to 8 days later, in terms of shortness of breath, dyspnea, acute respiratory distress syndrome, beginning a few days later, require ventilations of the lung artificially to provide oxygen to the person. Now, one of the things that we've noticed is, of course, during the beginning of the pandemic, that elderly were more susceptible to it. So on the following slide, slide 46, I talk about elderly at higher risk for COVID-19 than younger adults or children. Before we go into that, the pandemic actually took a big toll on older adults. That led to higher level of social isolation, mental health issues, malnutrition, and worries about their finances. So there have been collateral damages, so to speak, in terms of mental and other stresses that the elderly faced, not just the concern or the impact of the virus. And high disease severity and mortality have been noticed in individuals older than 50 years old, especially in the elderly. They are common observations in COVID-19. Several biological mechanisms have been suggested for this. Why are elderly more susceptible or vulnerable to this? The immunescence in elderly individuals could be primary reason. The availability of naive T cells or the decrease in the regulatory T cells with aging puts the adults in a vulnerable situation in terms of not gathering or providing the defenses, immune defenses to tackle the insult from the COVID-19. Basically, seniors have less innate and adaptive immune cells. Furthermore, the protective inflammatory response becomes worse with each aging. In addition, elderly persons are most likely to have serious comorbidities. In contrast with the pediatric age group, a lesser number of cases and reduced fatality have been reported. So the immune system and the inflammatory responses are the main contributors to the difficulties that the elderly face when dealing with the coronavirus infections. And the next slide gives an example to that situation, even after we started the vaccination campaigns. On slide 47 is taken from a report from Washington State showing their 7-day hospitalization rates per 100,000 population, comparing to unvaccinated, the top red line, with the vaccinated group. Basically, COVID-19 hospitalization rates among 65-plus-year-old individuals are 3.6 times higher in the unvaccinated population than in the population those who have completed the primary series. And again, this is data from Washington State Department of Health, and it is sort of is consistent with the in terms of the relationships or trends that we see vaccinated versus unvaccinated hospitalizations from other states in the United States as well. So vaccinations do help, and they significantly help, and is sort of important to our arsenal to fight off this pandemic. Now, along to this line of thinking, the next slide slide number 48, is actually a very recent Lancet article publication by Watson et al. Looked at various using statistical techniques as well as available administrative data on mortalities by adjusting for various factors, seasonal, demographic, and the country-specific approaches to report their data to see how many deaths we might have globally averted by vaccination. And basically, if you look at this, I apologize for the granularity of this original figure. It's granular. It's I tried to get it from the article, but that's the best I could do. But the top, if you look at on the left panel here, left side of the panel, it shows that the number of deaths that are averted is in between the red line and the gray line. Is considerable, and they have the best estimate of 2 million deaths. 20 million deaths have been averted by vaccination. So it's a large number of deaths that have been sort of averted by vaccination of the populations. And again, multiple countries have been used to generate this information. Now, the second thing they looked at, they said, well, the vaccines have been available mostly in more economically well-off countries, and lower-income countries might not be doing that well. And then, as the right panel tries to dig into that, their conclusion is, yes, the biggest savers of lives it can be higher in the higher income countries, but considerable number of lives also been saved in the lower income countries as well. So continuation of our vaccination rates and also targeting our vaccines to the new mutations are going to be our best way of fighting this disease, potential disease, and pandemic. Now, the vaccine of the coronavirus has not been staying still, and we had the Delta variant earlier on. Now we're up to a different Greek letter. Next slide, 49, shows the current variant, the Omicron variant, and this has turned out to be the most transmissive one, and it's making it very contagious. And it's comparing with this figure on slide 49, comparing with the old virus with the current virus and why it makes it so contagious. The left side, the blue virus, shows the spikes, and the spikes are, first of all, are considerably greater numbers, and they've also changed their shapes. Some of the Omicron's mutations form extramolecular bridges between the two spike subunits. They are shown in light blue. And the dark blue shows the comparison between the molecular bridges and the dark have the bridges. The bridges keep subunits from separating separating So this is very important. In the old virus, when the virus locked into the ACE2 receptor, some of the fit was not perfect and strong enough, so the the spikes split without getting hooked into the receptor, namely causing entry to the human cell. But the new variant, unfortunately, has reinforced itself through these molecular bridges, making it more strong in terms of hooking up to the human ACE2 receptors. All right, so what can we roughly say about how contagious or transmissive this virus, Omicron virus, is? Next slide 50 talks about that. Why Omicron virus is the fastest virus in human history. That's sort of eye-opener. Before Omicron, measles was the fastest spreader. It infected 15 others within 12 days. However, when Omicron's 50 or so mutation occurred around November 2021, it jumped from person to person very quickly. A single case could give rise to 6 cases after 4 days, 36 cases after 8 days, and 216 cases after 12 days. In contrast, the person with flu will give the virus to an average only 1 to 2 individuals, 2.3 individuals. So we're talking about dozens or hundreds of potential individuals affected by an infected person. This is the so-called epidemiologic modelers that have been using to predict the cases, etc., for the different variants of the virus. This is the raw variable is sort of the variable that they call it in their model is developed from these real-world situations in terms of the spread of how fast the virus is spreading. And biologically, the virus in its latest subvariant, BA.5, changed in 4 many ways. 3 of these alterations allowed it to evade human immune system and become infections, like we talked about earlier, whereas the 4th may have led it to produce more mild disease. So we got something to our advantage out of these mutations. Mutations in the Omicron led to slim molecular bridges that hold the subunits together, better for cell entry without breaking off, as we mentioned. Now, COVID's still causing havoc around the world. I mean, this variant, BA.5, accounts for about 80% of the new cases in the U.S., according to the U.S. Centers of Disease Control and Prevention, CDC. According to WHO, about a third of the new cases reported last week in Europe, a third in the Western Pacific region, and 25% in the Americas. The variant is keeping experts on their toes because of its ability to evade immunity and cause reinfection. So now this is a new concern that's emerging right now. The subvariant, BA.5, is dominant and causing more reinfections than earlier strains based on new analysis. And this could be happening for a range of reasons, such as mutations to BA.5 and waning immunity. Adding that, statistically speaking, you're more likely to get reinfected the longer it has been since your last infection, just based on the data that's been generated. So, continuing on this thought on slide 51, a biological perspective of some of these findings or observations. Previous variants, why is Omicron highly contagious? The previous variants 1 to 3 amino acids on the receptor binding domains were mutated. Altering each RBD, or receptor binding domains, just enough to prevent some, but not all antibodies from recognizing it. Whereas Omicron harbored 15 receptor binding domains that allowed it to avoid many more antibodies. However, when Omicron drastically altered its form to hide from the immune system, those changes eliminated some chemical residue that spikes need to attach to the ACE2 receptors in the humans to cause further infections. The final fourth change to Omicron led to a surprising weakness, exposing it to vulnerable to our bodily innate immune system defenses. While Delta was masterful in subduing interferon response, Omicron actually activated interferon response. Since lungs have more interferon response, this prevented it from spreading deeper into the lung and thus not causing severe illness. So this is the biological explanations or potential reasons for why we're not seeing that much serious infections and diseases from it. So now we are dealing with Omicron, we're dealing with coronavirus, and the end is not near in the sight. So we have to protect ourselves against COVID-19. So this is a bit of segueing to risk management, aside from risk assessment. So what do we have to do? The first one, as I mentioned, is vaccinations. Clearly the mRNA vaccines have been very helpful, and there are other vaccines also shown to be very efficacious as well. So when outdoors, when you go shopping and doing errands, I suggest wearing N95, if not, like I do, a KN95 mask, or a surgical mask if you have access to it. If not, a cloth mask with a PM2.5 filter inside for some protection. In the beginning of pandemic, I was doing just that. Was getting from online mask that has a pouch in it so I can put a filter inside it. So again, maintaining 6-foot distance is a good advice. And what we are observing in U.S. and many countries now, work from home if you can, so that you reduce your chances of contacting individuals outside your home that might be infected. And again, wear a good fitting mask always when indoors and in public places, especially crowded stores or gatherings, if you cannot avoid going into such large public area gathering locations. And I don't know how it is in Taiwan, but in the US you can do grocery shopping through online, and we do some of that, and some of it that we do it ourselves. So it's another way of trying to sort of reduce your chances of potential infections. Now, one thing I must note, though, this is based on a lot of experiences from my family and others, is that, as you know, airline travel has gotten very popular, very crowded, and quite a bit of hassles with also travel, but it's gotten too dangerous. I've heard too many people coming down with COVID after airline travel due to overcrowding terminals and, unfortunately, relaxation of masking guidelines. If at all possible, do not travel by air. Washing hands often to avoid transfer of virus to your eyes and lips, etc., is always advisable. If you're sick, stay in a separate bedroom from others as much as you can. And we have access in this country, and I think everywhere now, home antigen tests to determine your positivity or negativity status for COVID as your symptoms change. If you have fever, consult your physician to find out if you should be examined, and if you should start on an antiviral like Paxlovid or Remdesivir. So these are top of common sense things and also recommended things, I'm sure, across the board in many countries. Now the next slide deals with, okay, what do you do if you're at home or in an indoor location? That's slide 53. So reducing exposures to SARS-CoV-2 through building ventilation. There used to be an old adage saying solution to pollution is dilution, but this is now going to be sort of the similar theme for a viral agent that we are trying to protect ourselves. We spend about 80% of our day indoors, so maintaining high indoor air quality is essential. Of course, a large percentage of the time we're at home, either doing our daily routines or going to bed. Large droplets greater than 100 microns will settle on surfaces within seconds. In contrast, particles greater than 10 microns settle in minutes, whereas particles less than 5 microns stay suspended for hours or even days. So the settling can be interception, electrostatic removal, or diffusion, which takes much longer time for smaller sized particles. So the masks actually work on these similar principles too: interception, electrostatic deposition, and diffusion reductions. So coronavirus particles, as I mentioned before, except from humans, through cough and sneeze, can travel and be found in 3 different size categories, even though the virus particle itself is around 0.1 micrometers. So how do you approach indoor viral particle concentrations in terms of reducing them? Increase introduction of outdoor air. That's the most straightforward one. Use basically opening windows, etc. Use fans to increase the distribution of air and the effectiveness of open windows. If you're at home, you can adjust your HVAC systems, or actually, schools, universities, HVAC should be operated at the maximum capacity. And they should be inspected frequently. And then these systems use normally various filters that they have efficiencies, MERV 13 or higher filters, if they can be used by the design of the HVAC system, are recommended. And for closed-in spaces, including homes, like schools, small clinics, etc., it's highly recommended to use high-efficiency particle air HEPA filter air cleaners. And those HEPA filter air cleaners with HEPA filters can remove about close to 99 plus percent of the COVID coronavirus particles. In my home, I have a fairly large air cleaner that we run in our living room, dining room area. Well, with that, I think we should think about closing our lecture here. And the slide 54 gives my acknowledgments again to kind invitations from Janice Chan and Jasmine Chow. But at the same time, I wanted to recognize a lot of this work will not have been done unless many researchers from various institutions, not just EPA and companies like ICF or TMU, but universities, Harvard, and many other institutions and government agencies have collaborated to this topic. So the informations that I've gathered from various sources from these institutions have been quite helpful. Now, two last things. First of all, references that I cited in my presentation are provided at the end of these slides in the copy of my lecture presentations that you should have. So the file of my presentation has probably extra 4 more pages that has the references that are cited in my presentation that I went. And I really welcome any questions and comments if you have any. And I provide you with my email address here, haluközkaynak@gmail.com. So feel free to contact me, and I wish you all the best and luck in your future careers. Thank you. Goodbye.
And cut.